Greetings to all the fourth semester botany students. I'm Dr. K.Y. Pratibha, and in this session I want to revise what we had studied so far in the Unit 2, that is Embryology of Angiosperms 1. We had studied about the contributions of the Indian botanist Dr. Panchanan Maheshwari and B.G.L. Swami. Dr. Panchanan Maheshwari is called as the father. He is regarded as the father of Indian embryology, and he was an eminent botanist specialized in plant embryology, anatomy, plant physiology, and biochemistry. P. Maheshwari is noted for his invention of the technique of test tube fertilization of angiosperms and created a new hybrid plants. Another landmark discovery of Dr. Maheshwari was the production of haploids using anther culture. This discovery initiated a new era in plant biology. Professor P. Maheshwari was awarded Birbal Sahani Medal by the Indian Botanical Society in 1959. Another eminent Indian embryologist is Dr. B.G.L. Swami. Bengaluru Gundappa Lakshmi Narayana Swami was an eminent Indian botanist and Kannada writer. He served as a professor and head of the Department of Botany and principal of Presidency College, Chennai. Throughout his career, he has actively engaged in research work in the field of embryology of angiosperm. His contribution to the field of botany include discovery of vessel-less angiosperms, studies of monocot embryogeny, helobial endosperm, root stem transition. B.G.L. Swami was also given Birbal Sahani Award for his contribution in the field of botany in the 1975. So we have studied the structure of the typical flower in the Unit 2. Let us brush up what we have studied in the Unit 2. Flower is a reproductive structure of the angiosperm. The flower is the part of the plant that is responsible for making new seeds. The various embryological processes occur in the flower. That is why we have to study the structure of the flower. The flower consists of an axis, the thalamus or receptacle, bearing the essential organs, that is androecium and gynoecium, and also non-essential organs, the calyx and corolla. Typical flower consists of an outer whorl of green sepals that is called as calyx, which protects on the calyx. We can see the calyx consists of the units called as sepals, which protect the parts within. The second whorl has petals and which are brightly colored and are useful to attract insects to facilitate cross pollination. The second whorl is called petal, and it has individual units called as petals, and they attract the insects for the cross pollination. The third whorl of the flower consists of stamens, androecium, and are the male reproductive organs. Each stamen is made up of a filament, an anther, and a connective. Each stamen is made up of a filament and an anther and a connective. Each anther usually has two anther lobes. The anther produces pollen grains or microspores. The innermost whorl of the flower is gynoecium or pistil. It represents the female reproductive part, and it consists of ovary, style, and stigma. Inside the ovary, future seeds known as ovules are present. Each anther lobe bears. Now let us study the structure of the young anther. Each anther lobe bears two pollen chambers, also called microsporangia. Thus, dithecous anther consists of 4 pollen sacs or 4 microsporangia. That is why we call tetrasporangiate. Development of the pollen sacs begins with the differentiation of the archisporial cells in the hypodermal region below the epidermis at 4 corners of the young anther. The archisporial cells divide by periclinal division to give rise to a subepidermal primary parietal layer and a primary sporogenous layer. The cells of the primary parietal layer divide by successive periclinal and anticlinal division to form concentric layers of the pollen sac wall. So you can see here the differentiation or the development of the anther, when the anther primordia originates, epidermis will be formed, and then below the epidermis, homogeneous layer will be there. In that, archisporium will differentiate, and the archisporial cells undergo periclinal division, or the transfer division, to form the primary parietal cell and the primary sporogenous cell. And the primary parietal cell will undergo one more division to form the secondary parietal cells, which will form the endothelium, mid layer, and tapetum. Here, in case of primary sporogenous cells will form the secondary sporogenous cells, and the secondary sporogenous cells will differentiate themselves into microspore mother cells, which will undergo meiosis to produce the microspores. So you can see the development of the anther at the four corners of an anther. The wall layers from periphery to center consists of a single layer of epidermis, between which becomes stretched and often it protects. So single layer of epidermis, and it becomes stretched in the mature anther, and it is for protection. A single layer of endothelium with fibrous. Below the epidermis will be single layer of endothelium with fibrous thickenings, helps in dehiscence, and below the endothelium is 1 to 3 layered mid layer which stores food. Below the mid layer will be the layer of tapetum, and the tapetal cells may be uni, bi, multinucleate and possess dense cytoplasm. Tapetum is nutritive in function, which supply nutritive materials to the developing sporogenous tissue. The cells of the primary sporogenous layer divides further and give rise to diploid sporogenous tissue. A well-differentiated anther wall comprises of epidermis, endothelium, 1 to 3 mid layers, and the tapetum. Epidermis is outermost, protective, provides structural support, prevents the anther water loss, and they have the stomium region, which will help in the dehiscence of the anther to release the pollen. Endothelium develops fibrous bands of lignocellulosic secondary thickening that provides the mechanical force for the anther dehisce along with the stomium. 2 to 3 middle layers are short-lived and get crushed during pollen development. Sometimes it stores starch and gives nutrition to the developing pollen. Tapetum is very important. The fourth wall layer. So from epidermis, endothelium, mid layer, and the fourth wall layer will be the tapetum. Tapetum, fourth region of the anther wall will be the tapetum. It is the product of the parietal layer, and the tapetum plays a crucial role in pollen development. Cells are enlarged, radially elongated with dense cytoplasm, prominent nuclei, and they have all the cell organelles, endoplasmic reticulum, vesicles, mitochondria. Cells may be multinucleated or polyploid with rich DNA because of irregular mitosis and nuclear fusion. There are two basic types of tapetum, amoeboid and glandular tapetum. In amoeboid tapetum, the tapetum breaks down early at the pollen mother cell stage. So before the pollen grains are formed, only the tapetum breaks down. If the tapetum breaks down early at the pollen mother stage only, then we call amoeboid tapetum, and they are released to the protoplast and they grow in an amoeboid fashion. They encircle the pollen mother cell and they nourish the pollen mother cell to go into meiosis to form the pollen grains. In glandular tapetum, the cells remain intact and secrete the substances for pollen mother cell development. So they do not break down early. They wait till the microspores or pollen grains are formed. Once the pollen grains are formed, then only the glandular tapetum breaks down. Till then, they will be nourishing the pollen mother cells through their inner walls. The tapetum is involved in the functions of the tapetum. It supplies the nutrients to the developing pollen. It synthesizes callase to degrade the callose of the microspore tetrad, and it secretes sporopollenin, which will form the exine of the pollen grain. Tapetum forms the pollen kit. Tapetum forms the ubisque bodies, characteristic feature of the secretory tapetum, and and the tapetum is very important. It has certain the contribution of the tapetum is very important in the pollen grain. At the germ pore, the proteins which are very useful to the pollen grain in recognition of the compatible style is because of the tapetal contribution. And sterility of the pollen grains or the sterility of the anther is seen if the tapetum is non-functional. Okay, let's go to the next one. Mature anther contains similar layers as that of young anther with various modifications. So let us take up the structure of the mature anther. Epidermis is stretched and flattened. Endothelium loses water for dehiscence. Middle layers are collapsed. Tapetum is used up and sporogenous tissue developed into pollen grain. The stomium is the region of the anther where the dehiscence occurs along with the endothelium. So let us go to the next important aspect of the unit 2 is the microsporogenesis. What is microsporogenesis? The formation of microspores from a pollen mother cell inside the microsporangia of angiosperm through meiosis is called microsporogenesis. It comprises of the events which lead to the formation of haploid unicellular microspores. These directly functions as some of the sporogenous cells may directly function as spore mother cell and divide mitotically into a number of cells, and they may directly function as spore mother cells, or they may divide mitotically to form secondary sporogenous cells which functions as spore mother cells. Spore mother cells or pollen mother cells or microspore mother cells are diploid. All the pollen mother cells can form the pollen grains, or some disintegrate to nourish the developing pollen grains. All the pollen mother cell undergo the meiosis to produce microspore tetrads. The arrangement of pollen grain in a tetrad is due to specific type of cytokinesis. It is of following type. Simultaneous type in the cytokinesis occur only at the end of meiosis 2. The resultant tetrad will show the tetrahedral arrangement and is common in dicotyledons. Successive type in this type cytokinesis occurs twice, one at the end of the meiosis 1 forming the 2 cells, and then again at the end of meiosis 2 to form 4 cells. The resultant tetrad show isobilateral arrangement and is found in monocotyledons. So you can see here the pictures of tetrahedral tetrads. This is tetrahedral tetrad, isobilateral tetrad, and this is the decussate, T-shaped and linear. And this we can see the simultaneous cytokinesis, and this is the successive cytokinesis. There are 5 types of tetrads: tetrahedral, isobilateral, decussate, T-shaped and linear. Among this, tetrahedral tetrad is the most common. Let's come to the next aspect of unit 2, microgametogenesis. Microgametogenesis is nothing but the progressive development of the unicellular pollen grain into mature male gametophyte containing 2 male gametes and a vegetative cell. Pollen grain undergo first unequal mitosis to form a larger vegetative cell and a smaller generative cell, and is released in this binucleate stage, and after pollination it germinates through stigma and style. The generative cell then undergoes second mitosis to produce 2 male gametes or sperms in the pollen tube. The 3-celled male gametophyte has a vegetative cell and 2 male gametes. The pollen along with the pollen tube is known as microgametophyte. What is microgametogenesis? It is nothing but the development of single unicellular pollen grain into 3-celled male gametophyte is called as the microgametogenesis. A vegetative cell develops into pollen tube and carries the generative cell or 2 male gametes inside the style and style into embryo sac till the male gametes are discharged. So vegetative cell is very important. It differentiates into pollen tube once it lands on the stigmatic surface, and it carries the generative cell till the the ovule where the embryo sac are there, and then the pollen tube is involved in the discharging the gametes into the synergids of the embryo sac. Pollen wall will form outer sculptures called exine and inner thin intine during this process. Pollen grains pollen grains are normally spherical in shape. Each pollen grain has two layers, outer exine and inner intine. Exine is hard and made up of sporopollenin. At certain places, exine is either absent or very thin, and such places are called germ pores. Intine is thin and is made up of cellulose and pectin. A mature pollen grain has two cells, a vegetative cell and a generative cell. The vegetative cell is larger and has abundant reserve food material and a large irregular shaped nucleus. The generative cell is small, spindle shaped, and it floats in the cytoplasm of the vegetative cell. It is a cell within the cell. The generative cell in some cases divides to form two male gametes, thus making pollen three cells. In flowering plants, in flowering plants, pollen grains or microspore considered as the first cell of the male gametophyte. The male gametophyte is formed by microsporogenesis and microgametogenesis. Male germ unit concept consists of a stable association between the vegetative nucleus and germ cells, that is generative cell or sperm cell pair. This reproductive unit is transported within the pollen tube and delivered to the embryo sac as a coordinated unit. So let us go to the next one, abnormalities in the pollen grain, that is compound pollen grain and pollinia. What is pollen abnormality? The pollen grain shed, if the pollen grains are shed as aggregated as dyads, tetrads, polyads and many more are called compound pollen grains. Compound pollen grain may be an association of two, that is dyad, 3 that is triad, 4 tetrad, 8 octad, 12 or 13 or a large defined number of pollen grains, polyad. Wall cohesion exists in compound pollen grains. A pollinium is a coherent mass of pollen grains in a plant that are the product of only one anther but are transferred during pollination as a single unit. Example, Asclepidaceae and Orchidaceae. A pollinium, plural pollinia, is a coherent mass of pollen grains in a plant that are the product of only one anther and are transferred during pollination as a single unit. Example, I have told you Asclepidaceae and Orchidaceae. Abnormalities, Nemack phenomenon or pollen embryo sac. Pollen grains usually have two nuclei, but in some abnormal pollen grains, the number of nuclei may increase and also resemble an embryo sac. Hence it is called pollen embryo sac. So pollen grain behaving as embryo sac. Nemack in 1898 observed the embryo sac like pollen grains in the petaloid anthers of Hyacinthus orientalis, and he believed that they are formed by three successive division of the vegetative nucleus to form 4 to 16 nuclei, which may be organized like embryo sac or not. So it is called as Nemack phenomenon. Thank you.